Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 2nd March 2021. As we are into the revision mode, we are revising the entire syllabus in the form of CBSE sample papers. In a row we have done 4 papers, and right now we are into sample paper 5, 5th sample paper. In that, until yesterday, we have done section A, section B, and one question of section C. And as you know, section C consists of 3 mark questions. There will be 5 questions, 5 into 3, 15 marks. And in this, we are into question number 27. So here we go. Question number 27. What is the symmetry of a flower? Symmetry of a flower: actinomorphic, zygomorphic, and if there is, you can't cut a flower by using any vertical plane into 2 exact halves, then we say it is asymmetric, asymmetric, opposite to symmetric. Then we go to question number 28. In what kind of plants do you come across cranch anatomy? To which conditions are those plants better adapted? How are these plants better adapted than the plants which lack this anatomy? Now we go to straight the answer. Cranch anatomy is found in C4 plants such as maize, sugarcane. These plants can tolerate high temperature and high intensity of light. These plants are also adapted to live in the limited supply of nitrogen and carbon dioxide. These plants do not carry photorespiration. Unlike C3 plants, this helps in making the optimum amount of glucose. Hence, C4 plants produce more biomass compared to C3 plants. Next, we have got to go question number 29. Differentiate between chromatin and chromatid. So always differentiate. I advise you to put a table. So one side chromatin, another side chromatid. Chromatins are minute thread-like staining hereditary material found in the nucleus of the cell, whereas chromatid is defined as longitudinal half of each chromosome found during prophase. So one thread in a chromosome, as all of you know, there will be 2 threads, and if there is one thread, that one thread is chromatid. Next, second one, interphase and prophase. In interphase, it's called resting phase, but I don't like to use this word resting phase. Instead, I prefer to use this as preparatory phase. Just like 14th onwards, you have got your exams, 11th class final exams, and from today onwards or from the last 1 month, you are preparing for your board exam, annual exam. So it's something like an interface. So we don't call it resting, instead we call it preparatory phase of the cell, in which the cell is not engaged in the division, but this is important, DNA synthesis takes place. So that's why I prefer to call it as preparatory phase rather than resting phase. On the other hand, prophase, pro means first, phase means stage. It's the starting stage of cell division where condensation of chromatin occurs into short stumpy chromosomes. The nuclear membrane disappears at later stage of the prophase. Question 30. What are the inadequacies of 2-kingdom classification? Of course, we know it. The 2-kingdom system of classification is very much inadequate due to new developments and information about the organisms. Some of the inadequacies of these systems are 1. It is difficult to identify and distinguish the lower animals or plants because a few of them have common characters, like Euglena, a single-celled organism, possess a mixture of animal and plant characteristics. So it has a chlorophyll, but it is a heterotroph also. So naturally question comes, where to put it? Is it in animal or is it a plant? Question 2. Bacteria are microscopic organisms having a cell wall. They have prokaryotic cellular organization lacking nuclear membrane. They seem to be plants rather than animals because some of them are also able to synthesize food using solar energy. Third, the position of slime molds, chlamydomonas, sponges, lichens, diatoms is not clear in this system. Hence, two kingdom classification is a failure. You are finding it difficult to answer that question, then you can have the difference question. Define the term mycorrhiza. How are these associations affect a plant? Explain with example. Mycorrhiza is a mutually beneficial symbiotic association of fungi with roots of higher plants. So here it is the symbiotic association of fungi with roots of higher plants. Orchids grow as an epiphyte on plants, have their roots associated with fungi right from the seedling stage. Many orchids cannot survive without mycorrhiza associations. Many forest trees, such as pines, appear stunted if mycorrhizal associations are absent. In the presence of these associations, these plants absorb 2 to 3 times more nitrogen, potassium, phosphorus compared to without them. Hence, mycorrhiza is beneficial always. Now we move to section D, wherein it consists of 15 marks, and this 15 marks will be having 5 marks each, and there will be 3 questions. Of course, internal choice will be there for every question. So question number 31, what are the basis of classification of animal kingdom? Give detailed levels of organization. So basis of classification, it's the level of organization, symmetry of the animal or plant, layers of cells during embryonic stage, how many are there? Is it a diploblast or a triploblast? Then symmetry, is it symmetrical or asymmetrical? Presence of or absence of coelom, is it a coelomate or true coelomate, pseudo coelomate or acoelomate? Of course, metamerization, segmentation, presence or absence of notochord. So these are the basis for classification. And levels of organization, as we know, it is first cellular level, many cells join together, then it becomes tissue level, many tissues join together form then organ level, and organ system level is the highest. So in organ system level, higher and complex animals, various organs group to form a complex organ system like digestive system, respiratory system, circulatory system to perform specific function. Example, mollusks and chordates. You find difficult that question, then you got choice. What are the key features of Molaska? The key features of Molaska, of course, you know, freshwater. Mollusca are terrestrial or aquatic, marine or freshwater, having an organ system level of organization. They are bilaterally symmetrical, triploblastic, 3 layers, and coelomate animals, they have coelom structure. Body is covered by a calcareous shell and is unsegmented with a distinct head, muscular foot and visceral hump. A soft and spongy layer of skin forms a mantle over the visceral hump. The space between the hump and the mantle is called the mantle cavity, in which feather-like gills are present. They have respiratory and excretory functions. The anterior head region has sensory tentacles. The mouth contains file-like rasping organ for feeding the animals. It's called radula. They are usually dioecious and oviparous with indirect development. So these are some of the key features of phylum Molaska. Question 32. Describe various forms of lipids with a few examples. Lipids, that is fats, can be divided into 3 main types: simple lipids, compound lipids, derived lipids. Simple lipids, esters of fatty acids, They're called simple fat lipids. They can be further divided to fats and waxes. Fats, they are esters of higher fatty acids with glycerol. Waxes, esters of higher fatty acids with alcohol are called waxes. And second one is compound lipids. When esters of fatty acids with glycerol and alcohol contain other groups also, they are called compound lipids. They can be further divided into following: glycorophospholipids. As the name suggests, they contain phosphorus group. They are also called phospholipids. Example, lecithin, cephalin. And then sphingolipids, when phospholipids contain glycolipids. As the name suggests, these contain monosaccharides. Example, cerebrosides and gangliosides. And last, third one, derived lipids. When lipid is composed of hydrocarbon ring, what is important here is ringed structure, and a long hydrocarbon chain, it is called derived lipid. Example, cholesterol. Now, you find it difficult, then you can go for the choice. What is the choice question? Formation of enzyme substrate complex, ESC, is the first step in catalyzed reactions. Describe the other steps till the formation of product. An enzyme catalyzes a reaction. I told you an enzyme is like a cricket umpire. He will not do batting, nor bowling, nor fielding, but he will ensure that game goes on in a specific direction and complete the game without any problems. Here also, enzyme will ensure that rate of reaction will be fast and expected products are formed within the time. But enzyme itself will not undergo any changes. So an enzyme catalyzes, that's why we use the word catalysis. It means it hastens the rate of reaction. It speeds up the rate of reaction. Thus, if under normal circumstances, a particular number of units of products is formed, one can get many times more units in the same time when the reaction is enzyme mediated. An enzyme mediated reaction can be broken into three main steps, which are A. Formation of enzyme substrate complex, ESC. Transient stage, formation of the product. So you got three. In the first step, the enzyme binds to substrate at active site. This results in formation of enzyme substrate complex. Transient means in between. This is altered structure state of the substrate. This is the unstable stage. Had there been no enzyme, the required activation energy would have been much higher. But because of the presence of the enzyme, the reaction takes place with a less amount of energy. Formation of product, final one. Once the transient phase is over, final product is formed. With this, the potential energy of the system also comes down drastically. This can be shown by the following graph. Once the product is formed, the enzyme becomes free to bind with another substance. Look at this. Once the product is formed, now the enzyme is not required. Means it can go for the next one. So question number 33. Why is closed circulatory system more efficient than open circulatory system? In open circulatory system, the blood is pumped into set of arteries from where it flows into the large body space and bathes the various organs. It takes place in snails and insects, arthropods. That is, the blood will be like a swimming pool. There won't be particular pattern of flow because blood is not flowing through arteries and veins. That's why we say open circulatory system. Whereas in closed circulatory system, the blood remains enclosed within the arteries, veins and capillaries, and heart is the main pumping station. It is found in higher animals and is more efficient because the blood flows far more rapidly in closed vessels than in wide and open channels and body cavities. It takes much shorter time to circulate through the closed system and return to the heart. This quickens the supply and removal of materials, that is waste materials, to and from tissues by the blood. In a closed system, the arterial diameter can be regulated to alter the blood flow through them. So the volume of blood flowing through the tissue or organ may be regulated according to its needs by controlling the contractions and relaxations of the smooth muscles of arterioles. No such regulation is possible in open circulatory system like in cockroach. In case you are finding it difficult to answer that, then you can go for the next one. Choice question. Why is it necessary to check the RH factor of the blood in a pregnant woman? Is there any need for that? To check the RH factor in a pregnant woman. So what is the answer for this? RH is an antigen. It is present on the surface of erythrocytes, that is RBC, in about 80 to 85% of the human beings. That means if we see the RBC, over the RBC, if there is a protein, extra protein called RH antigen, then we say the person is RH positive. In case this RBC surface doesn't have this RH factor, then we say he or she is RH negative. So the individuals who possess this antigen are called RH positive, and those who do not have it are called RH negative. A person when exposed to RH plus blood develops anti-RH antibodies. Pregnant women who are RH negative, if bears an RH positive fetus, will develop anti-RH antibodies during the first delivery when the fetal blood comes in contact with her blood. The antibodies linger in the blood for sufficiently long periods. If she carries second fetus, that means if the lady becomes pregnant second time, then the RH antigens which are there in the body, the anti-RH antibodies in her blood enter the fetal circulation and cause damage to fetal RBC. That means the anti-RH antibodies which are present in the mother's blood will travel to the fetus, and in the fetus there is negative one, RH negative one. So the anti-RH antibodies will kill that, and as a result the baby which is going to be delivered will have to die because of the contrasting factors in the blood. So the mother may deliver a dead baby. That is the reason why the RH factor is a must when person, particularly RH positive and RH negative people, are going to get married. So this becomes fatal, means very dangerous. This condition is called erythroblastosis fetalis. Fetus is getting dead. That's why we say the fetalis. Erythroblastosis because the RBC will be blasting erythrocytes, and the second baby particularly will be in very danger. So first baby somehow can escape. That is the reason why we should consider this RH factor before marriage of any two couples. So this is how you need to answer.